So, can you see my screen?
Yes, I can see.
Okay, perfect. Sorry about that. Cher jury, chers collègues, chères familles, chers amis, j'ai le plaisir de vous présenter aujourd'hui les travaux que j'ai réalisés dans le cadre de ma thèse, intitulée Tomographie optique 3D multimodale, traitement spatio-temporel, correction du front d'onde et classification automatique. Donc, j'ai choisi de faire mon introduction en français et je poursuivrai ensuite la présentation en anglais. Donc, j'ai réalisé ces travaux à l'Institut Langevin sous la direction de Mathias Fink, Claude Bocara et Kate Gray. Donc, dans ma thèse, je me suis intéressé à imager des milieux dits diffusants. Nous avons un exemple ici avec la vallée de Yosemite. Si on regarde au premier plan, ici, on peut voir ces arbres sans aucun artefact. On est dans un cas dit de propagation balistique de la lumière, c'est-à-dire que la lumière n'a subi aucun événement de diffusion lors de son trajet entre les arbres et le capteur de l'appareil photo. Un peu plus à l'arrière-plan, ici, on distingue derrière un léger brouillard le half dome. On est dans un cas que l'on appelle la diffusion simple, c'est-à-dire qu'en moyenne, tous les photons ont été diffusés une fois depuis le half dome vers l'appareil photo. Et enfin, complètement à l'arrière-plan, on est dans un cas qu'on appelle la diffusion multiple, où les photons ont été diffusés en moyenne plus d'une fois, et où il est ici, dans ce cas, impossible de faire l'image de cet objet qui se trouve derrière ce brouillard. On peut modéliser très facilement ce type de système, constitué de petites gouttelettes d'eau ici, sur une épaisseur de taille L. Et donc, le cas numéro 1, dit balistique, correspond à un cas où, je l'ai dit, la lumière s'est propagée sans avoir été diffusée lors de son trajet. Il existe une grandeur qui permet de caractériser un petit peu les milieux diffusants, qu'on appelle le libre parcours moyen de diffusion, qui correspond à la distance moyenne entre deux événements de diffusion. Dans le cas numéro 1, on a donc un libre parcours moyen qui est bien plus grand que l'épaisseur du milieu. Ensuite, le cas numéro 2, je l'ai dit, correspond à un cas où finalement on va avoir en moyenne un événement de diffusion, mais il se peut que des photons ne soient pas diffusés, ce qui fait qu'on arrive à avoir une image finalement superposée dans le brouillard. Et le cas numéro 3 correspond à un cas où on a eu plusieurs événements de diffusion et où il est finalement très difficile d'imager l'objet derrière ce brouillard. Donc dans ma thèse, je me suis intéressé à imager essentiellement des milieux biologiques. En voici ici un exemple avec mon doigt éclairé en transmission par une LED. Et ce que vous pouvez voir, c'est qu'à la surface de mon doigt, on a une intensité qui est essentiellement homogène. On n'arrive pas à distinguer les structures qui sont dans mon doigt. Pourtant, à l'intérieur de mon doigt, j'ai un os qui a un absorbeur optique, mais on ne peut pas le voir ici. Donc, de la même manière, on peut modéliser ce système en mettant ici un absorbeur dans un milieu diffusant. Et puis, on peut regarder comment se propagent les photons. Celui-là, il peut être absorbé. Celui-ci, il peut, par exemple, passer le milieu diffusant. Si on veut comprendre réellement ce qui se passe, il faut avoir plutôt une approche statistique, c'est-à-dire répéter un grand nombre de fois ces propagations. Et si on le faisait, on se rendrait compte que finalement, l'information de cet absorbeur, elle va s'effacer dans le milieu sur une distance dont la taille caractéristique est à peu près la taille de l'objet, à cause de la marche aléatoire lors de la propagation. Et en sortie, on récupérera une intensité qui est à peu près homogène. Et ça, c'est parce que le libre parcours moyen de diffusion dans un milieu biologique, c'est environ 100 micromètres. Et qu'entre mon os et la surface de mon doigt, j'ai bien plus que 100 micromètres, ce qui fait qu'en sortie, on a cette espèce de brouillard. Autre exemple, ici avec ma main, et vous pouvez voir deux choses. Au-dessus, ici, vous pouvez voir mon os, et ici, une de mes veines. Pourquoi, dans ce cas-là, on arrive à les voir? C'est très simple. C'est simplement que ces structures, elles sont beaucoup plus proches de la surface. Et si on refait le même procédé que précédemment, on va se rendre compte qu'on a un phénomène d'ombre projetée à la surface de ma main, et c'est pour ça qu'on peut ici, dans ce cas-là, imager ces structures. Donc tout ça pour illustrer en imagerie un phénomène très connu, qui est le compromis entre la résolution à laquelle on va pouvoir imager les objets et la profondeur à laquelle on va pouvoir les imager. Donc j'ai représenté ici sur ce graphe différentes techniques d'imagerie biomédicale avec l'IRM, l'imagerie ultrasonore, l'imagerie photoacoustique, la tomographie optique cohérente que j'ai utilisée pendant ma thèse, les techniques multiphotoniques qui font appel aux non-linéarités et puis la microscopie confocale. Et ce qu'on s'aperçoit, c'est qu'il y a une tendance générale où ces techniques, finalement, elles se répartissent selon une droite. C'est-à-dire que si on veut aller imager profondément dans un milieu, alors on va devoir rogner sur la résolution. Donc, l'idée, quand on fait de l'imagerie, ce serait d'arriver, par exemple, dans mon cas, à utiliser l'OCT pour aller sonder en maintenant une très haute résolution le plus profondément possible dans les milieux. Donc ça, c'est très difficile, voire impossible. Pourquoi? On l'a vu, c'est à cause de ce phénomène de la diffusion, qui fait que si on s'intéresse à la profondeur à laquelle on peut imager les objets, qu'on la dimensionne par le libre parcours moyen, on se rend compte qu'au bout de 2 ou 3 libres parcours moyens, il reste qu'une infime partie de ces photons simplement diffusés ou non diffusés qui correspondent au cas balistique, qui portent le maximum de l'information sur l'objet. Et les autres, en fait, ils vont se superposer et venir créer des artefacts. Donc, est-ce qu'on peut faire quelque chose? Ben oui, on va le voir. Donc ici, je vais représenter schématiquement un objet enfoui à une certaine profondeur Z0 qu'on souhaite imager dans un milieu diffusant. Donc on va voir qu'on va avoir 4 types de photons qu'on va pouvoir récupérer suite à une illumination de ce milieu ici en rouge. Premièrement, ça va être des photons qui vont avoir passé dans le milieu plus de temps que des photons qui auront interagi avec l'objet, ou moins de temps. Et on va voir que ces photons-là, en fait, on peut simplement les filtrer, enfin simplement, avec un filtrage temporel, comme si on les chronométrait et s'ils arrivaient trop tard ou trop tôt, on les rejetait. Le deuxième type de photons, ça va être des photons qui ne vont pas non plus interagir avec l'objet, donc qui ne vont pas apporter d'informations a priori sur cet objet, mais qui vont passer dans le milieu le même temps que des photons qui auraient interagi avec l'objet. Et ces photons-là, on va voir qu'il existe un moyen de partiellement les filtrer à l'aide de ce qu'on appelle du filtrage spatial. Tout simplement parce que ces photons ne sont pas revenus du plan focal. Le dernier type de photons qui va nous gêner, on va le voir, ça va être les photons bleus ici, qu'on appelle les photons serpentiles, qui vont subir de légers événements de diffusion, mais qui vont garder quand même un état de cohérence, qui vont pouvoir passer les filtres spatiaux et néanmoins créer des artefacts sur les images. Et on va voir que ce type de photons, on peut les corriger en utilisant des techniques d'optique adaptative, pour finalement récupérer simplement ces photons rouges qui nous intéressent. Donc moi, dans ma thèse, je me suis intéressé à imager des milieux diffusants, à implémenter des techniques de filtrage pour m'affranchir au moins en partie de cette diffusion et pouvoir aller sonder les milieux plus en profondeur. Et je me suis aussi intéressé à implémenter de nouveaux types de contrastes qui pourraient avoir un intérêt pour aller un peu comprendre la biologie sous-jacente des échantillons. Voilà. So now I'll switch to English. So in the first part of my presentation, I will explain the optical modality I was using during my PhD, which is called full-field optical coherence tomography. And then in the second part, I will show you that by using the same setup, we can compute a new type of contrast, which we call dynamic full-field OCT, which is really useful to image living cells and tissue. And then in the third part, I'll show you how I try to bring this technology for in vivo retinal imaging. Okay, so the first implementation of optical coherent tomography was said to be time domain. So it's implementing this time gating I was talking about before. So the experimental setup is very simple. You have a source of light here, a photodetector, the sample is here, and then you have a reference. So why do we need a reference? So let's say we wanna have some sort of timer on the photons to reject them if they're too late or too soon. So if we want 1 micrometer resolution in a tissue, it would require a time resolution of around 10 to the minus 15 seconds, which translates in terms of frequency bandwidths to 10 to the power 14 hertz. So this is order of magnitude faster than the fastest electronic we have today. So we cannot really do this, let's say, numerically. So OCT rely on low coherence interferometry to, for a given position of the reference, observe on the photodetector interference pattern between the sample and the reference when the optical path length will match between both arms. And then when you want to do 3D imaging or 2D imaging, you just need to scan across the sample to generate those 3D image. So when you scan the reference actually like this, you'll get what we call an A scan, so it's just a line in depth inside your sample. And the actual resolution on this A scan is given by the coherence length, which depends on the spectrum of the source. Now, on the photodetector, we can write the electric field as the summation of the reference field and the backscattered inside the sample. And of course, we are measuring the intensity and not the electric field, so we need to square this expression so that some terms, which we call incoherent terms, will appear. And the information about the sample structure is inside those terms. So in order to get those terms, usually people rely on phase shifting interferometry by applying some phase shift using the reference, or by tilting the reference arm so that you can really physically separate those different terms. And with OCT, you can really get rid of those green photons there and filter them. So in terms of performance, here I'm representing the lateral PSF, so that the transverse resolution is of course limited by diffraction, so it's lambda over the numerical aperture squared. And the depth of focus, which correspond to the depth on which you're going to maintain this transverse resolution, is also given by the diffraction, and it's lambda over the numerical aperture squared. So that you can write this depth of focus in function of the transverse resolution. You can plot it, and then usually OCT is able to get a signal as deep as 1 millimeter. So if you want to get your A scan for a depth of 1 millimeter, you will be restricted in the transverse resolution of around 25 micrometer. So this is a first trade-off. So of course you could just translate the sample at the same time, but then it gets very slow. So you always have a sort of a compromise with time domain OCT. So to overcome this issue, Emmanuel Beaurepaire in the team of Claude Bocara proposed in 1998 another approach of OCT, which they called full-field OCT. So the main difference is that in full-field OCT, we use a camera rather than a photodetector so that we can parallelize each measurement on each pixel of the camera. So because we have 2 million pixels, it's a lot faster, and we get what we call unphased image, which correspond to an image at a given depth inside the sample. So now for it to work properly, we need to use a spatially incoherent source of light, such as an LED, so that adjacent pixel cannot give rise to interferences on the camera, which we call crosstalk. And we can arbitrarily increase the numerical aperture because there is no adverse effect on the depth of focus because we are only collecting signal at a given depth. Now we can also write the intensity on the camera like this, and we also have this incoherent part of the light and this interference term that we wish to extract. And in the same manner as OCT, we rely on phase shifting interferometry by using a piezoelectric translation on which is mounted the reference mirror to shift the phase here and reconstruct the image in post-processing. So with full-field OCT, we also have this time gating to get rid of those green photons, but in addition, we also have a little bit of spatial filtering because of this spatial incoherence of the source, which creates a sort of confocal gating on the camera plane. Now, in the second part, I'll explain how by using the same system, but that we can compute a new contrast, which is really useful to image living cells and tissue. So there exist many 3D label-free optical imaging techniques, such as second harmonic generation, which will give you pretty nice signal on fibers, for example, muscle fibers or collagen fibers. There is also the third harmonic generation, which will give you a high contrast on bilipedic layers. There is also the coherent anti-Stokes Raman scattering, which is sensitive to the chemical bond energy, so it will give you a contrast based more or less on chemical components. But all of those techniques require a very intense illumination because they rely on nonlinear processes and light matter interaction, so that you can because you need to have such high energy, you can only get signals from one tiny spot. So if you want to have a 3D volume, then you need to scan over your whole sample, and it's very long basically, so that the time dynamics you can access are really low. But if you take a typical tissue, you have kind of pretty fast dynamic inside. You have the blood flow in capillaries, you have the active transport in cells, the organelle motion, etc. And all of those processes happen at different timescales that can be quite fast. So the idea with dynamic full-field OCT is to make an image based on those motion and fluctuations. So if you try to image a living tissue or a living cell, there's no reason why the interference pattern will remain constant. Because if you have some scatters moving inside the coherence volume, then it can create some amplitude shift or phase shift. And with dynamic full-field OCT, we wish to measure those fluctuations. So how can we do it? So Clément Appelian proposed in 2016 to, rather than taking one or two images to reconstruct this full-field OCT image, was to take a whole movie. And then by analyzing this movie, we can see different types of behavior on different pixels here. Now, my goal was to quantitatively treat those different behavior to generate what we call a dynamic image. So here we have a movie of around 500 images, and we want to go to just one image. So how can we do it? So the first step is to remove the mean in each signal so that we can get rid of these incoherent parts. Then we have, again, these types of signal, and we want to go from these 512, in this case, values to only 3 values to construct a colored image. So I decided to do it in the HSV color space because it's really orthogonal to the human perception, so that I can really put some physical properties in each channel and then understand the image. So the first property, which was interesting, was the speed of the fluctuation. So in order to retrieve this quantity, I compute the power spectrum density of all of these signals, and then I extract the mean frequency, and I map it from blue for slow fluctuations to red for fast fluctuations. Then the saturation, I compute it as the inverse of the frequency bandwidth of each of those power spectrum density, so that white noises will appear grayish, basically just to improve the overall image quality. And finally, the value, which is the perceived intensity in the image, is computed as the running standard deviation over this signal, so that it will give an information about the amplitude of the fluctuation. So as an example here, if you take the raw data, so here it's very bright, remember, because of this incoherent part. You take a substack, and then on this substack, you compute the standard deviation, and you see appearing some structures, but it's very noisy. But at the end, when you average all of those standard deviation, you obtain something highly contrasted here, for example, on a mouse retina. So you have your 3-channel use, saturation, and value, and then you can recombine them to generate what we call the dynamic image here of a retinal organoid. So these retinal organoids are engineered by our colleague in the Vision Institute, Olivier Gouraud and Sacha Reichmann, and they're really useful to understand the development of the retina, for example, but also to do some disease modeling and drug screening. So on this image, if you have a bright area like here, it means that the amplitude of the fluctuation was high, whereas the color here, for example, blue, means that the fluctuation were slow, whereas here, red means that the fluctuation were faster. So now, thanks to the optical sectioning capability of dynamic full dosity, we can go in depth inside sample. And if we compare to the regular full dosity, we see that we have a much better contrast on cells. And on top of that, we can exhibit different types of behavior between cells, only based on their intensity, brightness, but also on their color, because they have different type of dynamic inside. Then, of course, we can reconstruct 3D volume to study the geometry of the sample. Now, if instead of doing 3D, you're interested in doing just a 2D image, but in a time-lapse fashion to see the evolution of the sample in the same plane, you basically face a problem. That is, the sample can drift. Because we have an IXR resolution of 1 micrometer, the slightest motion of the sample will switch the plane, and then you don't know really where you are. So I proposed a very simple algorithm based on the cross-correlation. So basically, I'm just looking and maximizing the cross-correlation during the imaging session. I could image the same plane for several hours here, another retinal organoid, where I could see a very interesting phenomenon such as cell division, cell apoptosis, and cell differentiation. And again, if we compare with regular full dosity, we see that we have a much better contrast on cells, and we have much more information. For example, we can access the power spectrum density and see different types of behavior. So far, I explained this dynamic full dosity technique, which is quantitative, which is completely label-free. We don't have to add anything else in the samples. It's also non-destructive, so you can follow the evolution of the same sample over several weeks or months. And we saw that we have a metabolic contrast. Now, I had numerous other results, but I prefer to show you the limitation of the technique. So, what about the limitations of full dosity? Of dynamic full dosity. So the first limitation is that the signal-to-noise ratio is much lower than that of full dosity. So for full dosity, the signal-to-noise ratio is proportional to the square root of the sample reflectivity, whereas for dynamic full dosity, it's not anymore the sample reflectivity, but the scatter reflectivity. And because those subcellular scatters are small, they have a very low backscattering cross-section, so that the number of photons we probe coming from those fluctuations are really small. So on this fly-through on a macaque retina, so it's ex vivo, you see that the deeper we go, the lower the signals. So it's not that the dynamic are lower deep down, but it's just that the signals we probe are lower. It's even more visible on this cross-section there. And if you look at the photoreceptor layer here with the cone and rows, I had to stretch the contrast to reveal them. So let's make a very simple model of how dynamic signals are generated. Let's assume that we have a scatter inside the coherence volume with a constant velocity. So this should give rise on the photodetector, on the camera, this should give rise to a sinusoid pattern. Now, if we take into account the backscattering properties of, for example, mitochondria, and then we add the shot noise, we will observe something like this realistically. And now you see that if you try to compute the running standard deviation on this signal, you'll end up basically computing the running standard deviation over the shot noise. So knowing this, how can we improve the signal-to-noise ratio? So on this graph, I'm representing two distributions. In solid line, it's a pure Gaussian distribution, random noise. And in dashed line, it's the same Gaussian distribution, but I added a little bit of bias, as if there was this small scatterer moving inside the coherence volume. Then, if you look at both those curves, you cannot really distinguish which one is which. Even if you compute the standard deviation, you'll basically obtain the same statistics. Now, rather than computing the distribution, if you compute the cumulative sum of the distribution, in the case of a Gaussian noise, a pure Gaussian-centered Gaussian, you'll obtain something which is called a random walk. So the maximum is expected to follow a Rayleigh law, so that it should scale as the square root of the number of samples, basically. Whereas when you have a bias, the bias will accumulate on this cumulative sum, and you will reach far greater extremums. So the idea is that theoretically, if we compute the cumulative sum and retrieve the maximum, we should have much more signal to noise ratio. So I did that on the same macaque photoreceptor layer, and you see that I was able to reveal some photoreceptor here, in addition to removing some of the camera noise here, the raw noise, because this noise was in fact centered. So in order to quantify the gain in signal-to-noise ratio, I segmented the cells, and then I computed the average signal-to-noise ratio per cell, and I saw that there was a twofold improvement. So depending on the sample, the improvements could be between 2 and 3. Okay, so now the second limitation comes from the very high sensitivity of dynamic full velocity. Because basically we are sensitive to the phase. If during the acquisition, the sample is slightly moving, then it's going to create some artifacts, some fluctuations. So finally, the signal we'll measure will be the summation of our real dynamic signal we wish to measure and this artifact. But because the sample is so much more reflective than those scatters, these artifacts will be stronger than our dynamic signal, so that it's going to be buried in there. So even if you have a very stable system with a sturdy optical bench, you can still observe, for example, because of the AC, some artifacts. So here, for example, on the human lung biopsy, you can see those white spots in the collagen fibers, which corresponds to artifacts. So those are not real dynamics. Second example here on this cell culture, so those are fibroblasts. Those are very flat cells, so they act as a diopter, so you have fringes on them. And then if you have a small vibration, the fringes are going to move, and this is going to be picked up by the algorithm. And then at the end, you cannot really see through the cell. You're kind of blinded by those fringes. So in order to unmix those artifacts from the signal of interest, the idea is that the motion between different scatterers in the tissue is completely uncorrelated. Whereas this motion actual artifacts should be highly temporally and spatially correlated. So the first step is to unfold this 3D cube of data, our movie, to a 2D matrix in space versus time. Then I use the singular value decomposition in the hope that those artifacts are going to be picked up on a few singular vectors. So I did this on an acquisition without any visible artifacts here. I'm representing the temporal singular vector. And you see that we have very smooth trajectories that looks like sinusoid pattern. Remember, it was our model, our very simple model. But with artifacts, we also have this very smooth trajectories. But in addition, we have very high frequency ones, which should correspond to those artifacts. So I propose a very simple way to detect them based on the number of times each curve crosses 0. So you have a natural increasing because there is more noise in the high ranked singular values. So I can just remove the tendency here. And then by applying a simple threshold, I can identify the the singular vectors corresponding to these artifacts. Then I can remove the associated singular values, reconstruct 3D tensors, and compute back my dynamic image. So on the human lung biopsy, you can see that I can remove most of the white spots in the collagen fibers here, but I can also improve the overall contrast on the image. Here, on the first acquisition in vivo of dynamic velocity, so it was on the mouth liver, so the mouth was breathing, which was causing some vibration. Also, there was the heartbeat. I can reveal some cells here in the bottom left corner, and also remove most of the noise in this area. And finally, on the fibroblast, I can remove most of the fringes artifact and really see through the cells and identify single scatters that I think were mitochondria in this case, and I can even track them. So far, I showed you this quantitative level-free non-destructive imaging modality, which we call dynamic velocity. I explained that it will give you images with a metabolic contrast. And then I showed you the limitation and how I could improve the signal-to-noise ratio by using the cumulative sum, and also remove most of the motion artifacts by using the singular value decomposition. So with this improvement, we started to envision the possibility of doing those measurements in vivo, because in vivo you'll have motion of the eye, for example, that are going to create artifacts. But if we have a way to remove them, then maybe it's going to work in vivo. So in this third part, I will explain how I tried to bring this technology in vivo by first tracking the retina, then correcting for ocular aberration, because we will see that those aberrations will lower the signal, and finally by imaging at different depths inside the retina. So first, why would it be interesting to get dynamic images in the retina? So the retina is composed of several layers. So basically the light goes from the top here, goes through the retina, is detected by the photoreceptor there, and then the signals are processed by the neurons inside the retina. So it's called the phototransduction process. And then the information here, the visual information is sent to the brain via the ganglion cells. And many diseases can affect the behavior of those cells, and those diseases can lead to vision loss and blindness. And we think that by using dynamic velocity, we could probe a change in the metabolism of those cells and maybe detect early those diseases and maybe understand better how they work. So imaging the retina is already challenging in itself because basically the proportion of the reflected light that goes inside the eye and is reflected and goes back outside is tremendously low. So for example, the ganglion cell layers, the proportion of the reflected light is approximately minus 80 dB, which means that if you send 100 million photons inside the eye, it will only collect back one photon, sorry. Okay, so why is that? Just because basically the light is detected here, so those are really transparent, and because the light is detected here, you have no reflection or no backscattering or not much inside the photoreceptors. So in order to bring dynamic velocity in vivo, basically we need three steps. We need to acquire images reliably in the same plane. Remember, we need a movie, like 500 images. Then we need to maximize the signal inside each image because we saw that signal-to-noise ratio was really important. And finally, we need to be able to get those images at different depths to probe different types of cells. So retinal imaging using full velocity was previously done by a PhD student in our lab, Peng Xiao, in 2018. So he removed the microscope objective from the sample arm here and replaced it by an eye. and it uses a near-infrared light source. So the problem with only this setup is that you need here to match the optical path difference between the layer you want to image with your reference arm. And because you have no feedback, it's very complicating by just using this full field OCT setup. So Peng had the idea back in that time to add a spectral domain OCT so that you could get a low resolution cross-section of the retina so that you could see where it was, and that could help him get the first images. So basically, if you do that, you'll see here, this bright area is the reference mirror there, and here it's a retina. So this is a real acquisition. It's slowed 3 times. This distance is 2 by 2 millimeter, and you see that the retina is moving quite a lot. So back in that time, Peng would click on acquire when both the retina here and the mirror were aligned. But you can guess that the probability of getting signals was pretty low because it was not actively tracking the retina. So then, in collaboration with Pedro Messe, a postdoc in our team, we implemented an axial tracking, an active tracking of the retina. So the first step was to average those B scans to get an A scan, here the red curve, and then to extract the displacement between each A scan, we compute the cross correlation so that we can really have a precise displacement, and we can, like it, control a very fast voice call motor to counteract the displacement of the retina and really lock a given layer with the reference arm so that we can image reliably the retina. So with this, we could get images with more than 70% chance of getting signals. It was pretty reliable, but we faced a problem, which was that the signal-to-noise ratio was not constant across the whole field of view here. So this was due to the optical asymmetry between the reference arm and the sample arm, because here we have a microscope objective. So we have a certain curvature of the coherence, what we call the coherence plane, and here we have the eye. And before we had 2 the same basically microscope objectives, so both of the curvature were the same, so we could get a nice and homogeneous signal. But now, because the curvature are different, we will only get signals where both are aligned, for example here. So a very elegant yet simple way to solve this problem was to add a glass plate in the sample arm. So how is it working? So if you have a scatter inside the retina, in the middle of the retina that is backscattering the light, it's going to exit the pupil as a plane wave orthogonal to the optical axis. Now, if you have another scatter on the edge of the retina, it will also exit the pupil as a plane wave, but with an angle. So that the distance both of these plane waves will travel through the glass plate will be different. And of course, because the refractive index of the glass plate is higher than the air, it means that it will add a higher delay with an higher angle, so that we can really have a various delay in the field of view, so that we can really make those 2 coherence plane curvature match together. So after calibration, we could get an homogeneous signal-to-noise ratio here on an LC subject on a 1 by 1 millimeter field of view, with cellular resolution up to 1 degree of the fovea. So this very wide 1 millimeter by 1 millimeter field of view acquired in a single shot is, to the best of my knowledge, the widest field of view that could be obtained by using an OCT with cellular resolution. So now, because of this wide field of view and this cellular resolution, we can compute very interesting biomarkers. So here I computed the cone density and see that for this LC subject, it was following what was in the literature. So with this, we reached our first step, which was to acquire images in the same plane reliably. Now, what about maximizing signal? So when you image an eye, in an eye, it's not perfect. There are aberrations, and we'll see that those aberrations will lower the signals, basically. And remember that those signals are really important, especially for dynamic full field OCT. So if you take a conventional microscope and you apply some defocus on here, a Siemens star target, you basically see that the image will get blurred. So this is known from decades because the optical transfer function of a conventional system is the autocorrelation of the pupil function. So in case of a defocus, in the pupil plane, we have a parabolic phase law so that we can compute the autocorrelation, get the optical transfer function. So here it's the modulation transfer function, so it's the absolute value of the optical transfer function. And then we can simulate what's happening. And you see here that when we defocus, we have a blur. Now, regarding full velocity, we have a different behavior. If you look here, for the same defocus, it seems that we have much higher frequencies. There is nonetheless some artifacts, some phase, some contrast inversion, and so on. But more importantly, there is a dramatically decreasing in the signal-to-noise ratio. So we really need to understand what is different between this conventional system and the full velocity so that we can correct for aberrations and get back our signal. So to do so, Victor Barol, during his PhD, made a mathematical model of the full velocity. And he found out that the optical transfer function for full velocity was in fact the autoconvolution of the pupil function. So it's rather different, especially for defocus here, because the modulation transfer function exhibits a much higher support so that we can have much more higher frequencies. But more importantly, the energy in this modulation transfer function is lower than before so that we lose, we have a dampening of our signal. So the first step was to experimentally validate this theory. So to do so, I built basically an adaptive optics full velocity. So this is the full velocity here. And here you have the adaptive optics loop. So you have a laser that serves as a guiding star, an adaptive element here to distort the wavefront, and a wavefront sensor here. So with this setup, I can acquire the full velocity image. I can also acquire the conventional image by blocking the reference. Thanks to the laser, I can get the PSF also. And by using this wavefront sensor, so this Shackartman, I can reconstruct back the wavefront. So for example, here, it's the combination of a defocus and an astigmatism. So then by running the previous model, so autocorrelation and autoconvolution, I can extract the modulation transfer function. And we can see that in the case of a conventional system here, we expect to lose those spatial frequency, whereas here, it should pretty much stay unaffected. So I can then do the simulations. So here it's the conventional image we're losing on those spatial frequency, which leads to a broadening of the PSF in the vertical direction, whereas for full velocity, the image is still pretty sharp. So the experimental data were really agreeing with the simulation. So I did numerous experiments. I also did experiments on single scatterers that were embedded in a gel. And with that, I could really validate this theory proposed by Victor Barol during his PhD. And it's going to probably be published soon. So then the idea was to use the same adaptive elements now to correct for ocular aberrations. So this adaptive element is a transmissive adaptive lens composed of 18 actuators, so that by individually controlling the voltage on all of those actuators, you can change their thickness, and then you can really bend the shape of the lens, so that you can really introduce some wavefront distortion, to counteract the ones encountered in the eye. So we only added this lens in front of the eye, so the system is still very compact compared to other adaptive optics approach. We do not use a strict pupil conjugation, and it does not require any calibration because we don't use any model decomposition. So this is called a wavefront sensorless approach because we do not monitor the wavefront, so we need to optimize on something. So I decided to optimize on the SDOCT, which serve, remember, at the tracking device. So I have this average A scan. And basically, I want for a given layer to maximize the peak height by penalizing the peak width, so that basically I can maintain this high XR resolution and compensate, for example, for dispersion and phenomena like this. Now, how can we optimize this? So I tested a bunch of algorithms, and the best one was the Dawn algorithm. So I don't have time to go into detail, unfortunately. But just know that this algorithm was really designed to handle noisy experimental data. So it's pretty robust and quite fast. So you can see here an example on a resolution target, and you can see that we have a 15-fold improvement, and even that at the end, the lens is able to correct for the tilts, which is very important for signal-to-noise ratio in full velocity, because the fringes are really spreading out. Then we can also look at the resolution, and you can see that during the optimization, we get back all of our spatial frequencies, and at the end, we have a pretty sharp image. So with this, we can correct now for all of those types of aberrated and scattered photons, and only recover most of the red ones. So I imaged one of my colleagues here on a dilated pupil. So you have much more high order aberration on a dilated pupil, and basically here it's the SDOCT, here is the unfast full velocity image, and you cannot really see his retina. So because the signal are so low, it's just the noise. But then after the correction, the aberration correction on the photoreceptor layer, we can reveal the photoreceptor. And here you can see that in fact there was this the vessel visible here. And then to our surprise, the correction was working for the whole field of view, 1 by 1 millimeter. But it's expected in the literature that the isoplanetary patch of the eye to be only 0.5 millimeters. So I have some hypotheses on why it's working, but unfortunately I don't have time to explain. But it's basically just a good fact. So now we reached our second step, which was maximize signal on each image. So the last step is to see if we are able to get signal from other layers. Okay, so now rather than optimizing on this last layer, the photoreceptor layer, I can optimize on the top of the retina, the nerve fiber layer, for example, here with the lens, and then I can start by imaging on the top of the nerve fiber layer, and here get a movie where when you average it, you can really distinguish the orientation of the fibers here, and you can see here a vessel. So if you go a little bit under, you can see this vessel going really inside the retina. You can, maybe not here, but you can a little bit distinguish the blood flow inside. And then if you go again below, it's in the area in between the nerve fiber layer and the ganglion cells. And I think those dark spots should be maybe ganglion cells. Okay, so with this, we see that we can image different layers in the retina, so it's really promising. So now it's time for me to conclude on this presentation. So in the first part, I explained how dynamic photodensity is working, so that it's a quantitative, label-free, non-invasive imaging modality with a metabolic contrast. Then I explained the different limitation of the technique and how I could overcome those limitations by implementing the cumulative sum and the singular value decomposition. And finally, I explained the different developments I made to try to bring this technology in vivo. So here is an example of a stitch image on the photoreceptors on another healthy subject. So as a perspective, we really now want to get this dynamic image. So there are still some challenges remaining, but I'm pretty confident that we could someday in the future get those images. I'm also interested in doing some computational aberration correction. So it was first done in full velocity by a German team led by Geraint Ottmann. So they were able to correct not with adaptive optics, but by using post-processing methods. And then it was also demonstrated by a Polish team led by Wojtkowski by using also a full velocity, but with an incoherent source of light, spatially incoherent. So it means that it should work. So this is a work in progress with an intern in our team, Cécile Maguin. And here you have a very preliminary result on a Tor Labs sheet of paper, where you can see before and after the numerical corrections. So there are some tricks, because theoretically we shouldn't be able to just to do it, but with some trick, I'm pretty confident we can do this aberration correction. And finally, I'm also interested in implementing a direct field version of the dynamic full velocity to remove part of the incoherent light so that we can increase even more the signal-to-noise ratio. So this was already done on a regular full velocity by Jules Scholler during his PhD in our team. So with this, he applied it on fingerprint imaging, and he could remove most of the specular reflection on the finger and image a lot deeper inside the finger. And finally, I would like to acknowledge everyone that was involved in this work, so especially my advisors, Matthias, Kate, and Claude. I would also like to thank the jury for agreeing on reviewing this work. I would like to thank, of course, the OCT team in the lab, also my office for the good atmosphere during these 3 years, and all of the Langevin Institute, the Paris-I group also here in the hospital, the member of the Vision Institute, so Olivier Gouraud, Sacha Reichmann, and Valérie Fado, for numerous discussion and giving us the sample to image. And finally, I would also like to thank my friends and family for their great support during these 3 years, and you for being here. So thank you very much.
Okay, so thank you for this broad and clear presentation. So you will have.
Juste le micro, juste le poser sur la table. We will. Can you hear me?
Yes.
You?